Bismillahirrahmanirrahim. Assalamu alaikum. Dr. Aisha Rashid here. I'm your instructor for the microbiology. Today we are starting with a new topic, which is control of microorganism, under which we will discuss various methods, principles, or various physical or chemical agents which are used to control the microorganisms. This discussion comes under the lecture series, lecture number 25. Before going to discuss the control or methods of controls of microorganisms, we must understand various terminologies regarding the control of microorganisms. The first term which we will see is antiseptic. Antiseptic is referred as a chemical substance that is applied to skin or the mucous membrane to prevent the growth of various kinds of microorganism, either by inhibiting the microorganism or either by destroying the microorganism. The point to be remembered here is antiseptic is used for the living things. Okay, we cannot say that this is uh, used on a table or a chair, any non-living thing. So antiseptic is specifically a chemical substance which is applied to the skin or the mucous membrane, mean which is applied to the living things. And what is the mechanism of this antiseptic? The mechanism is antiseptic. This chemical will either inhibit the growth of microorganism or it will kill or destroy the microorganism. The second term is disinfectant. Essentially, it is same as that of antiseptic, but this term is usually reserved for the use of eh, eh, inanimate objects. Inanimate objects mean non-living things, like I was talking about the floor, table, chair, or doors. So the chemical agent which is used to inhibit or destroy the microorganism or on inanimate or non-living things, which this is referred as disinfectant. Like Dettol is a disinfectant. We are using Harpic, or we are using different agents, phenyl, which is used for the floor. All these things are disinfectants. Then there comes the antimicrobial agents. Substances that either kills the microorganisms or prevent the microorganisms are called as antimicrobial agents. Under antimicrobial agents, there comes antibacterial agents, antifungal agents, antiviral agents, depending upon the kind of microorganism which they are affecting. So antimicrobial agents, substances which will kill the microorganisms. Then microbial agents, antimicrobial agents that kills the microorganisms. For example, microbicidal agents that positively kills the microorganism. Those are referred as microbicidal agents. These are bactericidal, uh, virucidal, fungicidal, indicate the type of microorganism they kill. So here is the difference between the antimicrobial agents and microbicidal agents. Antimicrobial agents will either prevent, inhibit, or destroy the microorganism, while the microbicidal definitely kills. There is no inhibition or prevention. Okay. So एंटीसेप्टिक और डिसइंफेक्टेंट में क्या फर्क है कि सब्सटेंस क्या है. एंटीसेप्टिक यूज होता है लिविंग थिंग्स के लिए, डिसइंफेक्टेंट यूज होता है नॉन लिविंग थिंग्स के लिए. ठीक है. अब एंटी माइक्रोबियल एजेंट और माइक्रोबिसिडल एजेंट में क्या फर्क है. एंटी माइक्रोबियल एजेंट्स जो है वो ऑर्गेनिजम्स की ग्रोथ को इनहिबिट करते हैं, रोकते हैं, या ऑर्गेनिज्म को किल भी कर सकते हैं. जबकि माइक्रोबिसिडल डेफिनेटली वो एजेंट्स हैं जो माइक्रो ऑर्गेनिज्म को किल ही करते हैं. ठीक है. The next term is sterilization. Sterilization, killing all the microorganisms present in any material, including the spores of the microorganism. This is very much important, and this is related to sterilization only. Sterilization not only kills the microorganism, but also kills their spores, which will become future microorganism. Under sterilization, microbiostatic agents are also there. The agents that nearly inhibit the growth of the microorganism, which totally inhibit, that merely inhibit the growth of microorganism, those as known as microbiostatic. As the name in the micro means microorganism, bio means life, static means stop. They inhibit the growth. Okay, these are called as bacteriostatic agents or fungistatic agents. Then next term regarding this is sanitizer. Nowadays very common. Any agent that reduces the bacterial numbers to safe level according to the pH requirement. In this corona pandemic, this is saying again, hand sanitizer must be used. So these reduces the number of bacteria, mean bacterial load on up to a safe level, means that number which do not affect. Now the conditions that affect antimicrobial activity. Some important variables to consider when assessing the effectiveness of antimicrobicidal agents are, when we are talking about microbicidal, means microorganism killing agent. So some important points are there which must we consider. First of all, when we talk about microbicidal, वो agents जो microbes को kill करते हैं, उनके मुतालिक important बातें. What is the size of microbial population? जिस targeted चीज की हम बात कर रहे हैं, वहां पे microbial load क्या है, microbial size population क्या है, ठीक है? So size of microbial population, intensity or the concentration of the microbicidal agent. What is the intensity? What is the potency? What is the concentration of that very microbicidal agent? Time of exposure to the microbial agent, either it is given 1 minute, 2 minute, hours. So time of exposure is also important. Temperature at which microorganisms are exposed to microbicidal agent. So temperature is also important factor. Nature of material containing the microorganism. So which type of material which we are talking about on which the microbicidal agent is used. Characteristics of microorganisms which are present. What are their characteristics? They are resistant, they are non-resistant. So all these parameters like size of the population, concentration and intensity of microbicidal agent, time and temperature, then nature and characteristic of microorganism. These are important. Now the mechanism of microbial damage. When we talk about all these things, microbicidal, microbiostatic, or whatever the agent we are using, then what is the mechanism, how the microbial damage happens? Antimicrobial agent either inhibits the growth of the microorganism or either kill the microorganism by damaging their certain structures. Like they will, antimicrobial agent can damage the cell wall. Antimicrobial agent can damage the cytoplasmic membrane or substances within the cytoplasm, such as antimicrobial agents can inhibit or destroy the enzymes. Antimicrobial agents can affect the ribosomes, or antimicrobial agents can affect the nucleic acid material of a microorganism. These are the mechanisms or the ways by which microbial damage happens. Now, the use of the temperature to control the microorganisms, to control the microorganism growth. The first one is method. There are various methods when we talk about the moist heat. For the moist heat, autoclave and instrument and equipment is there which can be used. For moist heat, boiling water can be used. For moist heat, another mechanism which is called pasteurization, that can be used. This moist heat has the ability to kill the microorganism. Okay, when we talk about the first method of moist heat, which is autoclave, in autoclave, the temperature is 121.6 degrees C at 15 pounds per inch square pressure. And at this temperature at pressure means 15 pound pressure and 121.6 degrees C temperature, 15 to 30 minute exposure will sterilize the various instruments various linings, various utensils like surgical instruments, and treatment trays, media like culture media, microbial media, or other liquids. So this is the speciality, this is the application of the autoclave. Autoclave applies 121 degrees C at 15 pounds. So sterilizing instruments. Then limitations. Infective agents, organisms in materials. These infective agents, sorry, autoclave. In fact, this autoclave is very ineffective against the organism in materials impervious to the steam, cannot be used for the heat sensitive article. So this is the limitation. Autoclave cannot be used, first of all, heat sensitive material, any material like plastics or like cotton or cloth, it cannot be used, autoclave cannot be used for the sterilization of heat sensitive materials. Moreover, the organisms which are also resistant to heat, to which heat is ineffective, those material cannot be used for sterilization in autoclave. Then boiling water. You know, boiling water temperature is 100 degrees C. Exposure of boiling water for 10 minutes, it will also kill the microorganism. It will kill the vegetative cells on the instruments and the containers. Usually in house, this boiling water is used for the baby's feeders or nipples. So it will kill the vegetative cells on instruments, on containers. Now, what are the limitations of this boiling water method? Endospores are not killed as in case of autoclave. So it cannot be relied upon the to sterilize. So we cannot say this boiling water completely sterilize any material. Then there comes the pasteurization. In pasteurization, temperature is 62.8 degrees C and the exposure time is 30 minutes. Or temperature, if temperature increased up to 71.7 degrees C, then exposure time will be 15 seconds. In pasteurization, it kills the vegetative cells of disease-causing microorganism. So vegetative cells are also destroyed by this mechanism. And it also destroys the vegetative cell or vegetative cell of microorganism in milk, in fruit juices, and other beverages. If you have focused on the various ad like Milk Pack, Nestle Milk Pack, or they say that this is pasteurized milk. So pasteurization is this very process in which high temperature is exposed for few minutes, like 71 is exposed for 15 seconds, and it will give even the vegetative cells of the microorganisms. Clear? So its limitation is that it does not sterilize. This is the autoclave picture of a steam autoclave. The functioning of autoclave is shown. This is the inlet point from where the steam enters, and here is the valve which controls the entry of steam. Either steam is entered abruptly or at high speed or low speed. So the valve to control the steam entry. Steam enters and it travels towards the main chamber. This is the main chamber where the objects are like instruments are placed. In it, in whole chamber, this hot air circulates and make it sterilized. After this, air is also exit from this very point. This is the exit through vent. It also travels through the channel. This is the steam exhaust where the steam leaves. Chamber pressure gauge. In the pressure is set from this gauge, we have set specific pressure, 15 pounds pressure is required. This is the steam exhaust valve. Steam is exit from here, and this it is controlled by this valve. This is the door by which the autoclave is opened and instruments are placed here. This is the picture of autoclave. You can see here the center, this is the door. This door is open and things are placed here. It is the temperature and pressure control buttons are here, and the instruments get sterilized. Now dry heat method. Dry heat is also a method for the control of microorganisms. For dry heat, hot air oven can be used. The temperature ranges from 170 to 180 degrees C and the exposure time is 1 to 2 hours. Now the application of hot air oven. Hot air oven is used for the sterilization of the materials impermeable to और damaged by the moist. हम पिछली स्लाइड में जो बात करके आए हैं moist heat, boiling water, autoclave, वो सारे materials जो hot moisture को sensitive होते हैं, moisture नहीं bear कर सकते, उनके लिए ये hot air oven use किया जाता है because in this hot air is moisture free. इसमें कोई moisture content नहीं है. Like it can be used for oils, for glass, for sharp instrument and the metals. Now the limitation of hot air, it is destructive to the material that cannot withstand high temperatures. कुछ drugs हैं, कुछ chemicals हैं, कुछ चीजें हैं जो high temperature नहीं bear कर सकती, वो destroy हो जाती हैं इस 180 degree C पे. Then there come the incineration. In incineration, hundreds of degrees C of temperature is used, like 1200, 1400. Sterilization of the transfer loops and needles, disposable carcasses of infected animals, disposals of the contaminated objects that cannot be reused. So this is used for the disposal, like mostly this incineration is used in hospital where the lot of waste material is there which is infected. Now the size of incinerator must be adequate to burn largest load promptly and completely. The potential for the air population exists because all things are burned. So this is the potential hazard for the potential hazard of pollution. Now low temperature. For low temperature, definitely freezers are used. Less than 0 degree C temperature is there. And the preservation of foods, it is used for the preservation of foods and other materials, and it keep them microorganism free. Mainly its disadvantage is mainly biostatic instead of microbicidal. Only it inhibits the growth for the time being when it is placed at 0 degrees C or less than 0 degrees C, but it do not kills the microorganism. Then liquid nitrogen refrigerators are also used, in which temperature is even minus 196 degrees C. Preservation of the microorganism happens. High cost of liquid nitrogen has to be beared. Now, the physical methods of microbial control. In physical method, there is radiation, filtration and desiccation. In radiation, 3 types of radiations are used: ultraviolet radiation, X-ray radiation, X-rays and cathode rays. Ultraviolet rays, X-rays and cathode rays. These 3 types of radiations are used. When we talk about ultraviolet rays, the action of these ultraviolet is the formation of thymine dimers. Thymine dimers will kill the microbial growth, will inhibit the microbial growth. Now, microbicidal effect is there with limitations owing to the lack of the penetration, reduces airborne infection in the hospitals. Ultraviolet rays are used to inhibit the airborne growth in various areas in hospital, in restaurants and in schoolrooms as well. Destruction of the microorganism surfaces in water also happens. Now X-rays, the process of X-rays by which the X-rays inhibit the microbial growth is ionization. Ionization happens in the microorganisms. Peroxidome formation is there which inhibit the growth of microorganisms. X-rays are usually used in research and research labs, used to induce the mutations. Then cathode rays, ionization also happens in case of cathode rays, and same is there, it is used for the research, may be used or for the sterilizing effects in the pharmaceutical houses and food industry as well. The second process for physical method is filtration. Separation of bacteria from the suspending fluid. Filtration mechanism or action is that filter separates the microorganism from the liquid from the fluid. Sterilization of the certain liquids which cannot be damaged by heat or the chemical treatment. Please wait for a second. I'm just coming. Okay, sorry for interruption. There is the filtration. Filtration is the next physical method for the control of microorganisms. In filtration will separate the microorganism or suspending microorganism from the fluid. Sterilization of the certain liquids. Filtration is used for sterilization of certain liquids which cannot be damaged, which can be damaged by the heat or the chemical treatment. Some fluids are there, like in pharmaceutical industry, for which we cannot use chemical agents, even which are destroyed by the moist or dry heat. For that fluids, this process is used. It separates the bacteria from the toxins, enzymes, measurement of the approximate size of some viruses. Then another process is there which is called as desiccation. It removes the water. It is effectively bacteriostatic preservation of various food. For foods, this desiccation is used. Here is the process of filtration. You can see here, this is the cup or the you can say funnel where liquid which has to be microbial free, that must be added. This is the membrane filter which has many pores. Then glass sport or platform is there which will hold this filter paper and cup. The base is there. This is rubber stopper to have the strong fitting with this funnel, this flask. When vacuum pump is applied, the liquid is sucked with the pressure into the flask, and all the microbes are remain on this one microfilter paper. Now, high temperatures. High temperatures are also used as a remedy to control the microorganisms. The use of high temperature is one of the most effective and widely utilized means of killing the microorganisms. Moist heat is much more effective than that of dry heat for killing the microorganisms because moist heat causes the denaturation and coagulation of vital proteins and enzymes of the microorganism. Usually when we cook the food, and we assume that by cooking all the microorganisms are killed because there is the denaturation or coagulation of proteins of microorganisms, and this proteins is very much essential for their functioning. When the proteins are coagulated or damaged by the heat, so microorganisms destroys, or there is the killing of microorganism. Moist heat will denature the cell proteins with lower temperature and shorter exposure times. Dry heat causes the oxidation of the organic constituent of the cell. And dry heat need longer time at high temperatures to achieve the same result. So this is the difference. Moist heat needs the low temperature with short exposure time, while dry heat needs high temperature with the high for long exposure time. Examples is there Bacillus anthrax endospores destroyed in moist heat. 2 to 15 minutes are required at 100 degrees C. For dry heat, you can see here are 180 minutes, great time, and temperature is also included. So that is why we are saying that moist heat is more effective. In it, exposure time is also short and temperature is also short. While in dry heat, exposure time 180 minutes and temperature 140 degrees C. Both are high as compared to moist heat. So vegetative cells, moist heat 5 to 10 minutes at 60 to 70 degrees C, while 5 to 10 minutes at 50 to 60 degrees C. Steam. Steam is another mean which is used to kill the microorganism. Use of pure steam under high pressure is the most practical and dependable way to apply the moist heat. It provides temperature higher than the possible steam without no pressurized and boiling. Rapid heat and greater penetration is there in case of steam. It make use of autoclave. Autoclave is its best example, which is very much popular lab instrument. Here is the table which shows the exposure periods or exposure times required for the aqueous solution for the liquids in various containers, affording a reasonable factor of safety for the sterilization by autoclaving. Now the container, if we talk about the test tubes, various dimensions, 118 into 150 millimeter, 32 into 200 millimeter, so various sizes of test tubes if we want to sterilize them. uh, in this case, minutes of exposure at 121 degrees C to 123 degrees C. This is we are talking about the autoclave. So the exposure time is 12 to 14 minutes, 13 to 17 minutes, and 15 to 20 minutes. If we talk about the flask, Erlenmeyer flask is there 50 ml, 500 ml, 1000 ml, and 2000 ml. The exposure time will vary from 12 to 35 minutes. Same is the case milk dilution bottles. If we talk about 9000 ml, it will take 50 to 55 minutes at 121 degrees C. Filtration and other mechanism of the sterilization or another mechanism of control of microorganism. Physical process, this is a physical process we use as we have discussed in last slide about the vacuum filtration assembly. This is the one way of sterilizing materials which cannot be sterilized by autoclaving or by dry heat in labs and industry. And we have discussed the reason if the chemicals or the materials or the drugs or the liquids which are sensitive towards the heat, either heat is dry heat or moist heat. For this, filtration can be used. Filtration uses the membranes, various type of micromembranes. Cellulose ester, these membranes are made up of cellulose esters of thin discs, are like 150 micron meter with the pores small enough to prevent the passage of the microorganism. So these microfilter papers or microfilter membrane do not allow the microorganisms to pass through. This filtration process is superior to older types of filters because pores of the membrane filters are of a uniform known diameter. The filter can be manufactured with any desired pore size. They absorb very little of the fluid being filtered, and the filtration through the membrane filter is more rapid than obtained with the older traditional filters. So it is also used for separating the types of microorganisms and for collecting microbial samples which are collected on the membrane. Efficiency is very high for this much high efficiency particulate air filter, HEPA filters, which are installed on the entrance of various rooms like in research labs, in hospitals. So these also filter the air. These HEPA filters are found in the biological safety cabinet, which trap the particulate matter such as microorganism. It captures 99% of the particulate matter from the exiting air. This is the filtration assembly. I have worked it a lot with my lab of pharmaceutical industry. So here you can see this is a machine or this is a vacuum motor. When switch on this, it will cause high vacuum. This vacuum is passed through this, it will causes the vacuum, and this liquid when passed through this, it will be filtered. Here are the filters installed at the mouth. Lyophilization and other technique which is used to control the microbial growth. Lyophilization causes the dehydration of microorganism, means it will you can see it will extract all of the water from the microorganism, and you know the water is very much essential for the growth. When there is no water, all microbial death will destroy and microbial life will destroy. So lyophilization causes the dehydration of the microorganism quickly at freezing temperatures and then sealed in container under vacuum. Osmotic pressure is also a measure which is used for the microbial control by plasmolysis. Plasmolysis means microbicidal effect in the preservation of various food. For example, in mats or high concentrations of the sugar, salts such as jelly and jams. So this one process is used. Now the control of microorganisms by using various chemical agent. Let me check the time. Okay. I guess we must discuss this control of microbial agents in next class. Otherwise, it will get long. Control of microorganisms by the chemical agents. So we will continue this discussion in the next slide. If you people have any type of query or any question, today's lecture was very much interesting. I enjoyed a lot. If you people have any type of query or confusion or your input or suggestions, you are most welcome to ask during live session. Thank you so much.